In this video, I want to talk about the cell life cycle and also get into cell division and the parts of it. So the cell life cycle actually consists of two different phases, right? And those are interphase and cell division. Now, interphase is actually the phase that cells will stay in for most of their life, okay? So this is the phase whenever the cells are just hanging out, they're producing proteins, they're living their lives, right? Doing what they're supposed to be doing. Now, when a cell gets ready to divide or make a copy of itself, then it will enter the next phase of its life, which is cell division, okay? And cell division really has two separate stages to it, okay? Those are mitosis and cytokinesis. So mitosis is division of essentially the nucleus, okay? So division of the nucleus and what is found inside of the nucleus, which we know is going to be the DNA within the nucleus. Cytokinesis is going to be division of the rest of the cell, okay? Essentially the cell cytoplasm. All right. So a lot of people like to think of mitosis as being cell division, and really it's not. Okay, it's only half of the actual cell division. Mitosis is just the division of the nucleus, the DNA, the genetic material within inside the cell. In order to completely divide the cell, we also have to go through cytokinesis, which is division of the rest of the cell outside of the nucleus, including the cytoplasm and all of the organelles in that cytoplasm. Okay? So let's talk about the phases of mitosis. All right, let's get into that. So there are 4 phases of mitosis, right? Those are prophase, metaphase, anaphase. I'm just going to come down here and do telophase. Right? So prophase, metaphase, anaphase, and telophase. Easy way to remember that is PMAT, okay, in that order. So let's talk about what is going to be happening in each one of these phases of mitosis. Now, before the cell enters into prophase, the first thing that it's going to have to do, right, before it can start division of the nucleus, is it's going to have to make a copy of all of its DNA that is found within the nucleus. Now, why does it need to do that? Why does it need to make a copy? Well, if we're going to be splitting that DNA in half into 2 new cells, then we need to make sure that we have full copies of the DNA for one copy to go into each cell, right? If you think about it in very basic terms, like say you have a blueprint to a house, and you want to give that same blueprint to 2 different carpenters in order for them to build 2 of the exact same house in 2 different locations. Well, in order to give each one of those carpenters a copy of the blueprint, you need to make a copy of the blueprint, right? You can't just take that blueprint and then rip it in half and give one carpenter one half and one carpenter the other half and expect them to be able to build the full house, right? You have to go and make a photocopy of that blueprint, and then you can hand each carpenter their own copy, and then they can go about building the house, right? So the same thing has to happen in your cells. You have to make a copy of the DNA, another copy of the blueprint, and then you give each cell its own copy. So that is going to actually happen at the very beginning of this whole cell division cycle. So before the cell ever even goes into prophase, it's actually going to make a copy of all of the DNA within the nucleus. Okay, so keep that in mind. Now I'm just going to draw a little simple cell here that is in prophase. So this is a cell, and then this inner circle is going to be our nucleus. Okay. So in prophase, one of the major things that is going to happen is all of that DNA that we just made a copy of is going to start to condense into structures called chromosomes. So before we enter into prophase, the DNA is going to be found in a structure called chromatin, right? Which really is going to look something like this. just a really big jumbled mess, okay? And if you were to look at that and I were to tell you, all right, take this DNA and split it in half perfectly right down the middle and give each cell its own little half of that DNA, that's going to be extremely time consuming and extremely hard to do, right? So during prophase, all of that chromatin is going to condense into structures called chromosomes, okay? So let me just draw those for you. So chromosomes look like little X-shaped structures, something like this. Now if you look at that compared to that last picture, it's much, much more manageable, right? It seems like it would be much easier to split these guys in half and get equal copies on each side of the cell. So again, these X-shaped structures are called chromosomes. Right, this is a condensed version of our DNA. So each half of a chromosome is called a sister chromatid. It's basically one copy of the DNA. Okay, it's each half. So these guys will eventually get split in half, and you will have one half go into one cell, and then one half go into the other. So we know we end up with identical copies in each cell. All right? So that's the first thing that's going to happen, that DNA will condense into these chromosome structures. We also have structures called centrioles that will divide and make copies of themselves, and they'll start to move to either end of the cell. Okay. Now these are eventually going to start to send out spindle fibers, all right, which will eventually attach to the chromosomes and will help pull those chromosomes apart as the cell divides. Okay, so that's another thing that's going to happen. Another thing that will happen is this nuclear envelope, right, that's helping kind of separate the inner contents of the nucleus from the outside will start to break down. It'll start to degrade or break down. And the reason for that is we need these spindle fibers to be able to reach those chromosomes in order to pull them apart. So it's important that that nuclear envelope completely break down. And in this next stage, you will see that it will be completely gone by the time we enter into metaphase. Okay, so that is the second phase here. So during metaphase, what's going to happen, the major thing that will happen is those chromosomes are going to line up down the center of this cell along a structure called the equator, also known as the metaphase plate. Okay, so those chromosomes line up, and the reason they're lining up is because it's going to make it nice and easy to split these guys in half. So remember these centrioles, these are centrioles here, are going to be found on either pole or either end of the cell, and they're going to be sending out spindle fibers, right? Spindle fibers are a type of microtubule that are used to help divide the cell, split the cell, and to also help pull chromosomes apart. Okay? So those spindle fibers will attach to the chromosomes that are lined up along that equator. And the centrioles, the microtubules themselves, those spindle fibers, will start to shorten, and they will pull on those sister chromatids of the chromosomes. And during anaphase, you will start to see those sister chromatids actually separating from one another. All right, so these spindle fibers are shortening, and you will see those sister chromatids getting pulled apart. All right, so the DNA is splitting in half, and each half is moving to either end of the cell. Okay? And then during telophase, this is the last phase of mitosis. So here the cell will really start to elongate. And you can see it starting to try to form two new cells, starting to kind of pinch off in the middle a little bit. And you will start to see two new nuclei forming. Okay, so that those nuclear envelopes will start reforming in each half of the cell. And inside of the nucleus, what do we know that DNA is going to do? Well, whenever the cell is whenever it's in interphase and it's just hanging out producing protein, the stage that the DNA wants to be in is the chromatin. Okay, so these chromosome structures will unwind and they'll start to form chromatin again, inside of these nuclear envelopes. Okay, so those chromosomes will start to unwind, and that will form chromatin inside of those nuclear envelopes. And eventually the nucleus will completely reform in each cell. And then after telophase, what do we know is going to happen? It was the very last phase of cell division after mitosis. It's going to be cytokinesis. Okay, so during cytokinesis, we will have complete separation of those cells. So essentially this double cell structure will kind of pinch off in the middle, and we'll have two new cells formed, two new nuclei, and we'll have all that chromatin found within those nuclei. And those two new cells that we have should be identical copies of each other. So we have identical copies of the cell that we started out with. Okay. So if you guys have any questions about cell division or mitosis, please don't hesitate to let me know.